Genetics and Embryonic Development, Module 3, Lecture 5. Development. The cranial cuttle is established early in embryogenesis. Dorsal-ventral is the second step. Finally, left-to-right axis is established. There are a number of primitive genes responsible for these processes. The HOX gene is a gene system that was first described in research in Drosophila. The HOX gene codes for a transcription factor. Drosophila or fruit flies have 8 HOX genes. Humans have 40 HOX genes. The organization of the HOX genes determines the anterior-posterior orientation of the organism. So here's just a diagram of the Drosophila HOX genes and then how many are in humans. And many are similar, but certainly not the same. But basically, the turning on and off of these HOX genes does determine anterior-posterior as well as dorsal-ventral. These genes are expressed in order. The position of HOXA and HOXB determines the head and the trunk. These genes have similar sequences in a variety of organisms, from fruit flies to humans. HOXD13 transcription factor. The normal HOXD13 gene transcription factor results in cell death in between the digits. A mutant form of this gene results in the not cell death, therefore syndactyly, or the fingers fused together. And this is an example of syndactyly. Programmed cell death is a common event during development, and certain developmental processes need to have cell death in order to have proper function. The TBX1 gene is located in the 22q11 region, and it's this TBX1 gene that is responsible for the congenital heart defects. Selective apoptosis or cell death is essential for normal heart development. When the TBX1 gene is deleted or mutated, conotruncal heart defects result. Here's an example of early embryonic development. And so the heart starts as 2 single tubes, and then it starts pumping, and then there are a number of curvatures that, so it starts as like 2 separate tubes, and then it turns upon itself. And again, there are a number of processes of cell death that's necessary to get it from these 2 tubes to the bending of a complete heart structure by day 35. So here we have the primitive heart, and then we kind of have outpouching the ventricle, then looping, and then separation into 4 separate chambers. And again, here are all the genes involved in these. You know, TBX1 is one of the initial steps. Then there's the induction signals, and finally TBX5, NK, and GATA4. These, to be honest, we're in our infancy of understanding all the genes involved in all the heart functions, but just to realize that there are embryonic genes involved. The nervous system the nervous system closes about 45 weeks post-conception. The CNS is actually built in waves of migration of neural crest cells. There are many genes responsible for neural migration. Holoprosencephaly is a very severe brain malformation resulting from loss of the LAS1 gene. Miller-Dieker is caused by a deletion of genetic material on the chromosome 17. The LAS1 gene is in the area that's deleted. As a result of the deletion of the LAS1 gene, the brain is smooth because the neural crest cells are not migrating correctly. So here we have, this is normal migration of all the different types of cells. And here we have Holoprosencephaly. All the cells are rather jumbled and they don't migrate correctly. The sonic hedgehog is another very important primitive gene. In mammals, the secretion of the protein of the sonic hedgehog, or SHH, by the notochord generates a gradient to organize the spinal cord and brain. The sonic hedgehog results in abnormalities. The sonic hedgehog can result in holoprosencephaly. When it does not function correctly, there's no right to left side. Here we have an example of Holoprosencephaly. This premaxilla is absent. This is the most severe form when we talked about in morphology, a single eye, and actually the proboscis is above. Here's a proboscis with a very close set or hypotelerism. Here again we have the cleft lip and palate without a premaxilla, and this is probably the mildest form. There's two nostrils, the eyes are close set. Hirshprungs disease is another example of neural crest disease in the gut. The RET gene is involved in proper migration of the nerve system in the gut. Lack of migration results in aganglionic gut, and the gut needs to use the nervous system for a peristalsis movement. So if the stool is not moving through the gut in a peristalsis movement, it gets stuck. The appendicular skeleton. Limbs originate from the lateral plate mesoderm. There are 3 signaling regions in the apical ectodermal ridge, or the AER. And again, most of these things were described originally in Drosophila. So here we have the AER, the proximal distal zone, or the ZPA. And here we have our HOX genes determining anterior-posterior. So in a certain sense, the limb, these are in the sonic hedgehog. Here's our sonic hedgehog. They're activated in a wave to result. First, the limb pops out, and then the digits come from, and then they grow with FGFR3, and then the digits. So when there is a mutation in one of these primitive genes, it can result in the lack of a limb. When it's unilateral, it could just be a sporadic event, maybe on the one side that there was a vascular defect or something, but when it's bilateral, it's usually an inherited gene. Many of these embryonic genes, they work for a very short period in development, but if they don't work, you have a problem. So there are many genes that we only turn on for 20 minutes in our entire existence during embryogenesis. That's the only time it's ever on. But if it's not on, if it's not functioning correctly, we end up with problems. So here are the genes associated with limb mutations, and there are a number of mutations within the system that can result in short limbs. Achondroplasia is one of the best examples. The fibroblast growth factor receptor 3, we used to think one gene, one disease, and life was simple, but unfortunately life's not that simple. In the fibroblast growth factor receptor gene, different changes within this gene can result in different phenotypes. In the case of achondroplasia, every single individual with achondroplasia has a mutation right here in the transmembrane region. Larger deletions in the middle of the gene or at the end of the gene can result in something called thanatophoric dwarfism, and thanatophoric means death-loving in Greek. And then there is a milder form of skeletal dysplasia caused by a different mutation within the FGFR3 gene. The fibroblast growth factor receptor 3 is responsible for chondrocyte differentiation and proliferation. So the ends of the bones are where growth is. So if the growth is not occurring at the ends of the bones, you get rhizomulic shortening of the bones. So in achondroplasia, we have rhizomelia, which is shortening here. The elbows don't usually fully extend, and there's a short hand held in kind of a trident configuration. So, question, the TBX1 gene is important in the embryonic development of the brain, innervation of the GI tract, the heart, the skeleton, or the spine. Question 2. What is the mechanism of action in the association of the development of the sonic hedgehog gene? Directly affecting gene expression, directly suppressing gene expression, concentration gradients, or suppression of transcription. This concludes this module.